Hello everyone, in this video we are going to talk about normal coagulation. Before discussing coagulation disorder in pregnancy, I thought I need to do some fresh knowledge about coagulation process because once we enter into specialty, we tend to forget basic physiology, and at that time when we start understanding the diseases, we got problem because of we don't know the basics. So this is for a revision purpose. So let's start. Coagulation means blood changes its consistency from fluid to gel. Hemostasis is a different thing. Coagulation is a different thing. Hemostasis means we need to stop the blood to get lost from the body. That means hemostasis. And what is coagulation? It change in the consistency from fluid to gel. Gel means clot. Okay. Now when we talk about coagulation, we have two particular processes occur simultaneously. Two particular processes. The first one is associated with platelets. There is activation, adhesion, aggregation of the platelets. If we talk about particular order, so first there will be platelet adhesion, then it will be platelet activation, and there will be platelet aggregation. The second process is fibrin generation. That means various coagulation cascades or various coagulation factors just do process, chemical process, and make the fibrin strands. Okay, deposition and maturation of fibrin strands. So two processes occur simultaneously when we talk about coagulation. Now, what is the purpose of coagulation? Is hemostasis in most of the cases. The purpose of coagulation is what? Hemostasis in most of the cases. Okay. Now, again, I have told you coagulation includes two specific processes: coagulation cascades and platelet activation. The goal of coagulation cascade is what? Is making a fibrin mesh like this, and the platelet activation and platelet processes is the plugging that particular fibrous fibrin mesh. Okay, now see, now let's understand coagulation cascade. What is the purpose of coagulation? Fibrin strands. Let's understand this very basic thing. We want here we want to make fibrin strand to make a mesh like structure. So fibrin is made from fibrinogen. When the fibrinogen is activated, it becomes fibrin. Now there is a thing called thrombin. Thrombin which will convert the fibrinogen into fibrin. So thrombin is the mainstay of generation of thrombin. A formation of thrombin is the main thing when we talk about coagulation cascade. Activation of thrombin is very important. Okay, thrombin is there. Thrombinogen we can tell. From thrombinogen, the thrombin is generated. Thrombin is an active form which will convert fibrinogen into fibrin. Okay. Now, activation of the thrombin is the main goal of cascade. There are two pathways has been described since years, that is intrinsic pathway and extrinsic pathway. One is intrinsic and one is extrinsic. Now let's understand why it is called, on the first hand, why it is called intrinsic and while this one is called extrinsic. When there is an intrinsic pathway, the cascade starts from the factors of the blood itself, while the extrinsic pathway, it occurs because of the injury. Cascade starts with outside injury and factors from tissues other than blood, that is collagen tissue factors, endothelium tissues. So this particular, there is a very clear cut difference between intrinsic and extrinsic pathway. Okay. which is very common, which is most of the time which is happening, extrinsic pathway. Okay, now let's discuss coagulation factors. Whenever we discuss about coagulation, we just tend to forget which number of the coagulation factor is which. So that creates so much of confusion while we are talking the pathways actually. So I have trying to make this video very easy. For example, these coagulation factors are what? They are basically enzymes, serine proteases. They are enzymes. What they do? They get activated by someone, and in turn they will activate others. It's very common thing, very easy thing. These enzymes, they get activated by someone else, and in turn they will activate other. Very simple. Now let's talk about one by one. Factor 1 is fibrinogen. Okay, now if I talk about factor 1 is fibrinogen, and factor 1a, this small a denotes activation, activated, it will be fibrin. Okay, factor 2 means prothrombin or thrombinogen, and when we talk about 2a, that will be thrombin, the activated form. Factor 3, tissue thromboplastin or tissue factor, which in sense will be doing extrinsic pathway. Factor 4 is calcium itself. Factor 5 is labile factor. Factor 6 is not now considered to be a factor because there is nothing like factor 6. Whenever the researchers were studying this coagulation pathway, uh, they were need to nomenclature some particular factor which makes some process. They name those factors by nomenclature, like factor 1. But in turn, when we go with again deeper research, we find that actually these factors are nothing but the molecules. For example, factor 4 is what? Calcium. It's very important for coagulation. And that's why the factor 6 was there to be considered earlier. But after knowing that the factor 6 is a part of some other factor, so factor 6 is not considered today. Okay. Now factor 7 is a stable factor. Factor 8 is anti-hemophilic factor. Factor 9 is called Christmas factor. Factor 10 is called Stewart power factor. Factor 11 is called plasma thromboplastin antecedent. Factor 12 Hagman factor. Factor 13 fibrin stabilizing factor. So there are 13 factors, and actually there are 12 factors because factor 6 is not considered nowadays. Okay. Now let's go with the pathway itself. This figure we have been seeing since years, since our junior days. Now let's discuss this thing. Okay. Now here is the intrinsic pathway, and here is the extrinsic pathway. What is our goal? Again, I told you our goal is to make fibrin. We need to activate the fibrinogen to convert into fibrin. This one is our goal. This is a fibrinogen, and this becomes fibrin. Okay. And which factor will do that? Thrombin. Thrombin is what? Activated factor 2. From prothrombin to thrombin it will happen. Okay. Now if we want to activate the thrombin, that is toward power we need to activate the thrombin. What we need a factor 10, that is toward power factor. Factor 10 associated with factor 5. So factor 10 associated with factor 5. Remember like this. These two activated factors are needed to activate that prothrombin into thrombin. Okay. So at this time, what we now need is we need to activate the factor 10. So When we are talking about external pathway, extrinsic pathway, there is trauma, there is an injury, and that injury will in turn make that factor 7 into activated factor 7. 7 into activated factor 7. That factor 7 along with tissue factor, which is tissue factor? Factor 3 is a tissue factor. Along with the tissue factor will directly convert or directly activate factor 10 toward power factor. Okay, and while we discuss about intrinsic pathway, it starts from factor 12. The 12 is activated, then comes the 11 is activated, then comes the 9 is activated, and then in turn it will activate 10. And when the factor 9 will activate 10, it needs the help of factor 8 also. Okay, the factor 8 is also get activated. Now what we get at the end, we get is fibrin. From fibrinogen we get fibrin. Now this fibrin needs to be stable. So the factor 13, that's fibrin stabilizing factor, will now in action and it will stabilize the fibrin. Okay, now this cross-link fibrin clot is very necessary for a platelet to clog them and to make a clot, a stable clot. Now plasminogen, you have heard this term plasminogen. From plasminogen it becomes plasmin, and that plasmin will do what? It will inhibit this fibrin. It will break these fibrin strands. Plasmin is there. Okay, so some plasminogen activator, if we have a plasminogen activator, then it will inhibit the clotting. Another inhibiting proteins are what? Antithrombin. Antithrombin will do what? It will inhibit thrombin and factor 10. So it will do anticoagulation. Factor C is also doing anticoagulation. See here. Active protein C will inhibit the here from factor 5 to activated factor 5, and here factor 8 to activated factor 8. So protein C will inhibit what? Coagulation too. Now this was all about cascade of coagulation factors. Now let's discuss about platelets. If you see here, this is a blood vessel and this is the endothelial cells, and these green things are platelets. They are just flowing with the blood. But once there is an injury here, for example, injury is happened here, and the collagen of endothelial cells is exposed, or blood vessel cells is exposed to the blood, and once it exposed to the platelets, the platelet is having basic three receptors. The main receptor is GP1b. GP means glycoprotein 1b. Other receptors are glycoprotein 1a, glycoprotein 2b. But the very important is what? Glycoprotein 1b. Okay. Now, this glycoprotein 1b directly cannot contact with collagen. It needs some stabilizing factor, and that factor is von Willebrand factor. What is von Willebrand factor? It stabilizes the platelet adhesion to the fibrin. So this all process I am trying to tell you is what? Platelet adhesion. Once it platelet adheres to the collagen, it gets activated. And this platelet activation is a very wonderful thing. For example, platelets have various granules in their cytoplasm. The major granules are granule alpha and dense granule. The granule alpha contains adhesion molecules, cytokines, chemokines, growth factors like that. And dense granule contains GTP, ADP, nucleotides, membrane proteins, and all other factors. So this degranulation will in turn activate the platelet more and more and will do a platelet aggregation. So the platelet will just come there and they will clog the fibrin strand and it will make a clot. So this is, I think, a very basic need for you is a needing the knowledge of coagulation. In next video I will tell about how to assess the coagulation with either PT and APTT. Okay, thank you friends.